Welcome to the Bioinformatics Q A session. We thought it would be nice to set this up as there's a decent cluster of interns working on bioinformatics related projects. This is the sort of first time we've ran these sort of sessions as part of the internship. Last week we had a Q&A for the Nimbus Cloud Compute, which we thought went quite nicely. And as it's a Q&A session, it's unstructured. So I suppose we'll just sort of dive into it, sort of invite people to come forward with their questions. And if everyone is, and we'll see how we go from there.
So does anybody have any burning questions that they want to ask? Otherwise, I'll start and get you. Basically, there's two things we can do. Either you can start asking questions, or I'm going to emotionally blackmail you by saying, can you all introduce yourselves and tell me what you're doing, which everybody hates. So them's your options. Which one would you like?
I'm fine with either one.
Okay. Maybe not everyone's a big introvert that feels awkward with these things.
How about we go with a brief introduction then? Would you,
all right.
Yeah, Sarah, do you want to pick? The order is different on every screen, so.
Yeah, well, the first person for me is Emily. You're an intern and not a helper? Is that?
No, I'm actually a mentor as well.
Okay, yeah,
so,
I wasn't sure.
Yeah, no worries. But my background is in astronomy, so this is kind of all new for me. I have no questions, but I'm very keen to hear all the answers.
That's a great way to live, I think. I guess I should also introduce myself, actually. I always forget to do that. So so my name is Sarah. I'm the Life Sciences Application Specialist at Pawsey. So I work in the supercomputing team. I actually did a PhD in neurogenetics and a postdoc. And now I help biologists and researchers get going on the supercomputer, anything from like training to these kind of Q&A sessions to optimizing their code, installations, like all sorts of different things, which is quite nice. I enjoy that it's a nice mix of technical and going out and talking to people. It suits me. So I'm really interested to hear what you've been doing. I did read some of the applications when we did the technical reviews, but I haven't been following along since. So I'm sure you're all doing some very interesting things. So maybe we'll start with Kevin to drop you in the deep end, since you've come up on the top left for me. Like what are you up to?
Cool. So I guess I've been getting more into it recently, so I can better say what I'm actually up to. The project is looking at essentially, like, I'll probably use a bit more terminology stuff, since most people here probably have like a biology background. So metastatic melanoma samples. So they've extracted that from patients and they put that into a mice, and basically we're sequencing those samples, trying to determine genetic markers of skin metastases.
Okay.
So there's going to be like a bit of processing steps with filtering out the mice genomes, and there'll be exome and RNA sequencing occurring. And that's sort of what I'm doing, the RNA-seq stuff now, using just a variety of different packages on the Nimbus instance I've been assigned.
Okay, cool. And so I guess with that, like, is it single cell data, or is it kind of
I don't think so. No, I think it's just everything.
Okay.
That's why we have to do all like the filtering steps as well once we've got the actual data, because there's a bit of mice RNA, a bit of human RNA. We've got to figure out what's what. And at this stage, I'm just doing trying to sequence the RNA, everything, and then we'll worry about the what's what later.
Yeah. Okay. Cool. That's very interesting.
Yeah.
And melanoma is always very relevant in an Australian context.
Definitely. My mom's a pathologist, so like her job is looking at human cells under a slide to determine if it's like cancer or whatever. So she's always telling me to put like my sunblock on, look after myself.
Yeah, absolutely. The health outcomes for advanced melanoma, as I'm sure you know, are not good. So prevention is important there. But that's very interesting. And so you're getting I guess, a different perspective from something that you've probably grown up with if she's been looking at pathology slides for a long time. Then
Yeah.
So
Yeah.
that's quite cool.
Great. Yeah, and there's 44 samples altogether, so that should be a lot of fun stuff to work with.
Yeah, definitely. And you can experience the joy that is bioinformatics workflows, which are
yeah.
can be
Yeah.
can present their challenges.
Oh, so far so good. I'm really enjoying it.
Oh, awesome. I'm really glad to hear that. And where are you based for your project, I mean?
Harry Perkins, so that's a medical research center in WA.
Oh, awesome. Actually, yeah, that's where I did my PhD and postdoc. Well, I actually worked there for 7 years, if you include like honors and being a research assistant and whatever.
Nice.
Yeah, I was with Nigel Lang and Gina Ravenscroft doing rare diseases upstairs on level 7.
I'm on level 6, so just a level below you. Well, below where you would have been.
Yeah, level 6 is the cooler level. They have a better lunch culture. So there you go. Okay, well, that's really cool. I'll move on to the next victim. How about you, Dina?
Yep, it's Dina.
Dina.
Yep. So the project I'm working on is, and by the way, I don't have any background in biology, so I'm sorry if I'm using the terms incorrectly, but the project I'm working on is trying to basically look at whole slide images and then use machine learning to predict potential patient response to different treatments to liver cancer.
Mm-hmm.
And yeah, so so far I have been trying to sort of write scripts to download the data from GDC and then sort of extract the patches to make them suitable for feeding into the network.
Yeah, awesome. That's really interesting. There's a lot going on in that, like machine learning with slide pathology type of space, it seems. So hot and relevant topic.
Mm-hmm.
And so where are you doing your project?
So I'm myself in Sydney, but I think the project is at Curtin University.
Okay, yeah, awesome. And so what's your like kind of technical background, or I guess you come from like a more comp sci, or?
Yeah, I'm doing software engineering and computer vision. So,
yeah. I mean, that's great. Like I personally feel that we need more of that type of cross-disciplinary action, right? It's really useful. Like I don't come from a software engineering background. I often wish that I did. So yeah, being able to get both of those things together is really good. Yeah, awesome. Well, that's very interesting.
Yeah.
All right, who else can I pick on? Maybe Joel?
Yep. Cool. How's everyone? I'm working at UWA for my project with Daniel, who's actually in the same room as me. He's sitting right next to me. So I guess I can talk on his behalf, or we can talk together. But our project
Feel
is
free to tag team.
Yeah, I'm tag over to him halfway through. But our project is on assembling genomes from a sample of, or 72 samples from a marine biofilm, which has been taken off the coast of West Australia. So our whole project is on developing a software pipeline and trying to automate that to taking the sequence data and splitting it into to sort of characterize the biodiversity of the sample and its like metabolic function as well. So how it works and sort of develop a picture on how the sample actually interacts with its environment, etc. So using a lot of stuff like quality control and sequence trimming software toolkits to be able to do that sort of metagenomic analysis. So that's pretty much it. Like we're trying to, like I said, automate it so that hopefully for future investigations, we can, for this similar sort of data, we can have less computational or less intense computational work. You can sort of just run it, and there's a lot of time that we have to wait to let the computer sort of do their jobs. It's all like there's a lot of data. It's like each file is 15 gigs each, and we've got 72 of them, so there's lots to work with. But no, we're making some good process, writing some scripts. I'll pass over to Dan, I guess.
Yeah, sure.
Yep, that's the project doing bioinformatics work on 72 biofilm samples. And yep, our work has just been throwing together scripts and automating everything, so we can get the data from it.
Okay,
Yeah,
and
so far we've just been using the Nimbus instances to do all the comp work on it. It definitely takes a while.
Mm.
Yeah, that it does.
Yeah, those steps can be quite computationally intensive. And I guess also if you're a Nimbus, you can't, because you don't have like a huge amount of computational resources, I'd imagine you can't like scatter gather or whatever, which you might otherwise do. Or just, it's probably, I can imagine it's probably like an embarrassing parallel problem if you have enough compute. You can probably run a lot of the samples independently. So so in terms of your approach to automation, like are you using something like Nextflow or SnakeMake, or are you like what's your?
So far I've just been using bash scripts to run just recurse through all the samples and do that. Nothing like Nextflow, which maybe it would be a good idea to use.
Yeah, I would definitely suggest thinking about it. I mean, obviously it's your project, but yeah, either one of those is probably a good thing, especially if you're only going to be there for 10 weeks and you're trying to leave like a product behind that the lab can presumably continue using. Yeah, Nextflow can be really good for that. It's also a good learning thing. It seems that Nextflow is becoming more desirable in other fields that aren't just bioinformatics, so I hear. Whereas SnakeMake seems to be a bit more limited. So if you're going to choose between two, it seems like Nextflow might be the smarter choice, but really, I mean, they're kind of the same thing. It's a different syntax to do the same thing. But I mean, starting off with bash scripts also makes a lot of sense because like you just need to make sure that something runs, right? And you're kind of getting the logic, and then you can worry about implementing that into Nextflow later if you felt like it.
No, thanks for looking into it. Sorry, I've got a really bad like echo thing going on.
Yeah.
I
think one of you will have to mute.
Oh, yeah.
But yeah, so the end goal is to leave something behind that the lab can continue using in the future. So that sort of tool we'll definitely look into, or any way that we can sort of make it more robust and make the whole process in writing these scripts easier and more complete, I guess. So yeah, thanks for that.
Yeah, no worries. I mean, it's something that we tell people quite often, and most bioinformaticians do start out with it's almost like your embarrassing dirty laundry of like these ugly bash scripts or R scripts or whatever, and then you have to like clean them up. The other thing is that's really good about Nextflow is it makes it a lot easier to scale onto HPC if they want to do that later. Also something to think about, because bioinformatics installations are always a bit awful, is containers, if you haven't thought about that already. So Nextflow interfaces really quickly with containers.
Yeah, because we want to build a container. We've got like too much software to not really. We want to build one to have all of our tools in.
Yeah.
Because sorry, Joel, I believe you're going to be transitioning from the Nimbus over to Photonics during your project. That's the aim. Oh,
okay, great. In which case, that's even more useful to have something like Nextflow, perhaps. Otherwise, you're going to have to there's going to be an overhead in getting all of your bash scripts Sloan ready anyway, I guess. Oh, cool. That's exciting. Transitioning from Nimbus to HPC in a short period is cool. So Yeah, okay. Who else can I annoy? Brittany, are you an intern or a mentor? Sorry, you're muted.
Hello. Yeah, I'm an intern.
Great.
So I'm currently at Murdoch University in WA. So I'm doing my PhD in plant genetics. So I'm a little bit more from a lab-based background. So my honours involved doing a GWAS on a large population of barley varieties to look for genes controlling the formation of stomata. So in the ag industry, we're quite interested in modifying the stomatal characteristics in crop plants, because reducing the stomatal densities actually improves the yield quite dramatically under drought stress. So that can potentially mitigate some of the economic losses seen across WA by like $180 million a year if we can modify the stomata in the way we hope to. So part of my project is lab-based. So I'm doing some gene editing experiments on some candidate genes that I found in my GWAS. So some virus-induced gene silencing to confirm function, and then we'll hopefully move on to CRISPR once the function is confirmed to do the knockdowns and make them stable. So we're hoping to develop some new lines that we can potentially commercialize in the next couple of years. The other phase of my project is moving a little bit more into bioinformatics. So we've got some new genomes that have been sequenced. I think it's like PacBio sequencing. So it's a lot more effective. They've done some, I forget the terminology for it, but it's like all the gene annotations have been confirmed through like RNA-seq, so they know that they're expressed. So it's really good data sets that we've got. So we've got 95 new ones that have come through. A lot of them are wild varieties as well, which will be really interesting to look at. So for my project, I'm going to be doing like a pan-genome interrogation for 100 pre-selected genes for stomatal development and functioning. So these are genes involved in like regulation of stem cell development in the leaves, and like, because the stomatal pathway is quite heavily regulated by that, circadian rhythm genes. So there's a whole bunch of genes that all interact with each other. And I'm hoping to run like a large scale analysis of those genes for all the different genomes that I'll be looking at. So at the moment, I've done some preliminary work. I looked for the core and dispensable genes across all the genomes, and not too surprisingly, because the stomatal network's quite conserved, but almost all of the genes are core genes, so they're present in all members of the pan genome. But at the moment, I'm waiting just for the annotations to come through, which will be this week, so that I can rerun that analysis on the 95 new genomes. So I've currently run it on the current 20 ones that have been sequenced. That was like 2020, I think they were released. So I'm waiting on those annotations to come through so I can rerun it. And at the moment, I'm currently setting up a small cluster on my Nimbus instance. So I've got a cluster of 3, I think it's a 64 RAM flavor. And I'm hoping to just speed up some of my processes a bit more if I've got a cluster running. And I'm just setting it up through a Slurm, I think a Slurm managed cluster. Yes, I'm currently working on that.
Yeah, wow, that's impressive. You've got a lot going on.
Yeah.
I guess also because it's kind of like forming part of your PhD, you have this broader context around it rather than like coming in for a discrete 10-week.
Yeah,
period.
that's right.
So yeah, that's exciting. Like, I come from a human genetics background, so I'm always really interested to hear more about the plant side, because I just didn't. I'm learning more about it now because helping different people. So that's really interesting.
Yeah, the annoying thing is that plant genomes are huge.
Yeah.
So, and like unlike human genomes, which have like that are heavily annotated, a lot of the time when you find candidate genes, you have to do a lot of further investigation, look at the domains, see the gene families, because a lot of the times it's not very clear what the gene does because the annotations are just not there. Yeah, it's a very new field, but really fascinating, and hopefully find some really cool variations between the different varieties for these genes that might be potential targets for, drought tolerance experiments in the future.
Yeah, I mean, it's very relevant, right? Like we are only going to become more reliant on this type of technology in agriculture, I think. So
yeah, exactly.
And the long
That's
read
definitely
picking
where
thing
it's
is
moving.
interesting. Yeah, I mean, it has to. I think.
Yeah, exactly.
Yeah, long read's also interesting.
Yeah.
Okay, so yeah, I mean, sounds like you're. I don't think I've got any suggestions. I think you know what you're doing.
Hopefully.
Yeah, seems legit. Yeah. Okay. Well, cool. Yeah. All right. Who
Awesome.
else
It's good to
can-
know I'm on the right track.
Yeah, I mean, like I said, I can't comment too deeply on the science, but
Yeah.
it sounds like you've got a good mix going on. Also, I also had like they have different terms: soggy scientist, humid scientist, this mix of wet lab and dry lab, which is bioinformatics and, being on the bench. And yeah, I did find that was useful. If nothing else, I found it gave me more credibility with wet lab scientists that have never done bioinformatics, because they're like, yeah, you can commiserate with like the terrible Western blots or cell culture, and they get the sense that you understand, which is true, because you are literally like crying in the cell culture room, like, why?
Yep.
Yeah, so I mean, that's I guess a soft benefit that might not be immediately obvious otherwise.
Yeah, exactly. Having that versatility there is really good.
Yeah, and the understanding as well. I think if you've never done it, can be hard to understand what's going on in the lab. So
yeah, exactly.
So yeah, okay. Well, cool. Very interesting. All right, who else can I be going? Abraham?
Yeah, hello.
Hey.
Yeah, well, I'm based at UWA, specifically in the Corrosion Center. So I'm very close to the Pawsey, just crossing the road.
Oh, cool.
Yeah, well, and my project is it's nothing associated with, biology or something like that. Specifically, it's about corrosion science, corrosion engineering. I'm applying machine learning and also a specific technique called Bayesian networks, which is basically artificial intelligence. So what I'm analyzing is the risk of a specific form of corrosion in oil and gas production called stress corrosion cracking. So, and for an alloy, which is duplex stainless steel. So I'm just analyzing the risk and the probability of this material if stress corrosion cracking could occur or not. But since you have, well, in oil and gas engineering, you can have a significant number of variables, not only the chemical composition of the material, but also the environmental parameters such as temperature, pressure, chloride concentrations, corrosive species, etc. So the point is analyzing all those kind of variables and then determine if stress corrosion cracking could happen or not. And essentially is because in industry standards, this material is extremely underrated, and the performance of these materials is overly conservative. Some of them, well, if you cannot apply this material, immediately you need to use some alloys which contain much more nickel, and that increase the cost significantly. So basically, we cannot run experiments. We can only check for information, for standards, for some other historical data, historical corrosion reports, and then we need to determine the probability of stress corrosion cracking. So basically, all the work behind my project in Pawsey has been gathering all that information and forming from zero and a data set. And when you are trying to combine several sources of information, the problem with that is that some research report certain parameters, such as, I don't know, concentration, pH, something like that. Some other are not reporting that. So you have a lot of significant missing information. So the work behind has been, applying machine learning, deep learning to perform some analysis of missing data imputation. And after that, we have used Bayesian network. We are using specifically software called BioLab, and we have trying to infer such volumes of missing information. And right now at Pawsey, I'm just trying to compare my model with another model, which is based on stream gradient boosting methods. It's a tree-based model, and we are trying to analyze the results, if they can be comparable each other, if we are missing some connections from my network, or if maybe I'm missing some points that the stream gradient boosting can read, because that's the thing with the stream gradient boosting. They can connect very efficiently all the variables and offer you several predictions. As long as you can, adapt or parameterize this model properly to infer all the data accordingly to the phenomenon.
Yeah, okay, that's interesting. So are you already embedded in this research group, or are you just new to this project, like as of a couple of weeks ago?
No, I've been in these research groups in the beginning.
Yeah, okay, yeah. So like you're doing a PhD, or?
I'm doing a PhD actually, yeah.
Yeah, okay, yeah, cool. I was gonna say, like, you know a lot, like you can't just have come in a couple of weeks ago.
No, yeah, I have been studying a lot of machine learning and artificial intelligence and deep learning. So yeah, I'm not new in the topic, but yeah, my time here in my internship is just for that specific purpose. And then I can continue with my research, and maybe I can finish it, because as I said, it's very impractical trying to run experiments using that material. And when you are analyzing all the phenomena with, in oil and gas, the main problem is that you need to use hydrogen sulfide. And hydrogen sulfide can be very poisonous, it's dangerous. As a PhD, I cannot work with that. I need a specific training. And every experiment requires more than 30 days. So it's a lot of time. Yeah,
that's tough. Yeah, well, I mean, you've been able to come at it from a different angle, and it sounds really quite interesting. I'm afraid, obviously, it's not really my field, so I can't offer you really any words of wisdom apart from sounds important and relevant. I know Western Power was doing something not really the same, but they were trying to predict which electricity poles were likely to like catch fire or fall over next. And so I guess, as you probably already know, it can be quite relevant to industry, and it's very interesting.
Yeah, absolutely. Yeah, since the beginning of my project, nobody has been capable of helping me substantially. So everything has been about researching and asking for the software and for the methods, and so everything has been for me very, very new since the beginning.
Yeah.
So
yeah, you're kind of on your own.
Only one of my friends, one of my supervisors is based or is totally focused on the mathematical framework. So from that, we have been capable of modifying the algorithms accordingly to obtain our results.
Have you ever visited the Perth Machine Learning group?
No.
Yeah, so they're based, it's kind of like a meetup group, but they have some good traction now, I think, where so they meet up fairly regularly, and it's a mix of like academics, industry people talking about machine learning, and they have training courses sometimes. So in bioinformatics, it can be the same thing, that you're what we call a lonely bioinformatician, where it's just you and nobody else in your lab basically has any idea what you're doing, because they're all wet lab people. And so if you need help with something, you're on your own, unless you have developed your own network. So yeah, they could be like a good place to start if you're a lonely machine learning person.
Okay, yeah, for sure.
Yeah, there must be a better term for that, but I haven't heard one yet. So okay, awesome, thank you. Patrick, if you're there. Oh, you can't use your mic. Oh, okay. So your project is on performing molecular dynamic simulations on a peptide called LFG, which has been recently discovered to improve the A and B antifungal drug. I'm using the simulations to better understand the structure of LFG in water, which is kind of the first step to seeing how LFG improves the A and B drive. Okay, cool, very useful. And molecular dynamics, I guess, is something that's well suited for computational work and HPC and that sort of thing. So that's nice. Bioinformatics, I think, is still kind of bridging that gap a little bit. So that sounds quite good. Thanks for dropping it in the chat. Yeah, I understand sometimes you can't use the mic or have the camera on or whatever, so no stress. Cool, and then last but certainly not least, I'm sorry, I'm going to pronounce this wrong. Jiayu?
Jiayu, yeah, Jiayu.
Okay.
Yeah. Hi everyone. I'm working in the University of Queensland, and my project is about model the structure of the Ebola virus delta peptide and the formed pore on membrane by using the GROMACS and visualize and analyze the result by using the visual molecule dynamics. I have obtained some experimental data through CD spectral TPLS, EIS experiments, which can be the reference for us to the simulation. And up to now, I just learned how to operate GROMACS and VMD and try some simple simulation to model the structure of the peptide in water box.
Oh, wow. Okay, awesome. So similar-ish kind of work to the to Patrick, perhaps.
Yeah, we are in the same research group actually.
Oh, okay, cool. Okay, awesome. Yeah, it's good that you can. It's nice to have someone else also working with you for the 10 weeks, or perhaps longer if you've been embedded there for a while.
That's so great.
Yeah. Yeah, so that's good. I mean, GROMACS again is like one of those very well optimized softwares that's well suited to this type of resource, which is really nice. Can get stuck in and do your thing quite not easily, because obviously it's very complicated work, but with fewer barriers perhaps than stupid software installations and that sort of thing, which can be a problem in bioinformatics, unfortunately. Okay, well that's yeah, it's been really great to hear a bit more about what you're all doing, and it's quite a diverse range of projects, which is nice. And also seems like a diverse skill set. Like not everybody's a biologist, there's some like comp sci type of people and, you know, a mix, which is great. So I guess, are there any other questions either of me or of the mentors or of each other that you might have? Anything to like technical to project-based to like career stuff to commiserations about things not working or stress or whatever, like, I don't know. It's a bit tricky over Zoom sometimes as well. Also, I don't know if you can hear my cat banging around in the background. She wants to be let out of this room, but I can't get up. So she's stuck in here with me, which I'm happy about.
I have been working on the project, and there have been like a few little things that I found that like helped me along the way, like running screen, for example. So if my my laptop shuts down, the commands are running in the background. And I've just tried to integrate VS Code into the Nimbus instance, and that seemed to have helped a lot too. I was wondering if you had any more of those, like, I don't know, little things that could help a lot when trying to work with either the Nimbus or the Cytonics instance, or just with the bioinformatic projects in general.
That's an excellent point. Yeah, so VS Code, we do actually have some documentation in our Confluence setup about how to set up VS Code for Nimbus or also for Cytonics. It's really useful. I use it like all the time. I've also converted other people. So I would definitely recommend giving that a go, because it's much nicer than just using like a plain terminal or something like that. Obviously there's like lots of different ones one could use, but VS Code's pretty good. Screen, so yeah, screen is also really useful. thing, because when you log out or go away or close your laptop or you get disconnected, your process is still running in the background and it doesn't get shut, which is really useful. God, what else? Definitely documenting what you do as you go. So if you think that you will remember in like 48 hours to 8 weeks time, you are sorely mistaken, my friend. I'm sorry. I also thought this for myself and then always would be very regretful that I didn't make better notes at the time. You won't remember. Basically write your documentation or write your comments as if you are writing it for somebody else, because you are somebody else in eight weeks or whatever. Yeah, definitely learned that one the hard way. Yes, I don't think, I don't know if Edric or Emily or anyone else might have any similar tips or tricks for this sort of thing. It's kind of hard to like come up with it on the spot. It's like, there must be loads of little things, but it's like, what do I actually do?
But I mean, like if you're working on a Nimbus instance, it's important to remember or know that you can do things like set up a Jupyter notebook instance if that's how you normally work.
And
so that's like
too.
Yeah, you can have or even like work through VS Code, right? Setting that up with your instance. You can have kind of all the creature comforts pretty much that you would normally have on your own local machine, just a couple extra steps. So yeah, don't let that be a major bottleneck to your productivity, because that can be for some people.
Yeah, it's much nicer to have something like that. Also, I guess I've heard, sorry, I'm just gonna let the cat out. She's really annoying. I'll be 2 seconds. Sorry about that. Thanks for waiting around. What was I going to say? It's gone now. Creature comforts, Jupyter notebooks. Oh yeah, I've heard from a lot of people that they're like, oh, I feel like a bit of a fraud because I just Googled the answer to something. Like, no, we all do that all the time. That's the right thing to be doing. Stack Overflow and that sort of thing is immensely helpful because most of the time it's a really common issue, or like the error message you've gotten is unhelpful, but someone else has definitely seen it before. So like learning how to get good at Googling and troubleshooting, I think, is a key skill to come away with. Yeah, and it's not always easy. Like it definitely takes time to develop the ability to be like, oh, okay, like this is the problem that's going on. This is the like suggested solution. I think I'm getting a vibe of like what's actually going on here and what I actually need to change, and digging around and fixing stuff. Yeah.
I like that you frame that as a skill you need to sort of develop itself. Because I know that when I started out working working on the command line, working in Unix, I'd go to try and find out how to install a piece of software and just see a bunch of just gibberish that I just could not even pass. I'd be looking at it, there'd be words, and but I couldn't see how they actually translated to, actions on my PC. So, and it's hard to define exactly what changed over time, but it's just like you build up your comfort and you have enough things to link together. Like I think the amount I have committed to memory is far less than my ability now to just like head to some documentation and find that information quickly.
Yeah, absolutely. That's so true. Even for simple, not that simple, like things like Conda installs, or like I've been struggling with a user ticket, trying to get this particular pipeline working for this group, and like, oh, it's very frustrating. But yeah, it's a lot to do with, it has all of these different Conda environments and stuff. And so being like, oh, what's actually like in this particular one and this sort of thing. Like I'm always going back to the cheat sheet or Googling, or like, how do I clean up all the packages in Conda? Like I can't remember because I don't do it every single day. So it's not about necessarily committing it to memory. Like maybe you would have to do, I don't know, in biology, you kind of do have to remember things, I guess. But it's more about having the flexibility to be like, oh, okay, well, I know what I need to find and I know how to find it. Yeah, actually, yeah, with my students, I would always say like one of the things you need to learn is how to troubleshoot on your own and troubleshoot well. And like it takes time. It's not something you just develop in a week. It's almost like a soft skill in a way. It's a bit intangible.
Cool, thanks.
Yes, long answer.
A question I have for you, Sarah, is sort of what's the key difference between something like Singularity and Nextflow? Like, I haven't worked with Nextflow before, just seen the term.
Oh, okay. Yeah, so Nextflow is, there's different terms for it, but like a workflow engine or workflow manager. So in bioinformatics, typically you don't just have like one software or one process. You have sometimes like 5 to 10 to 20 different job steps and processes all linked together in a chain or a workflow or a pipeline. And so piping the inputs and outputs between those in a reliable way that has checkpointing, that you can resume, that will tell you what versions of the software it used and this sort of thing. If you're going to write that yourself, actually, it's really a lot of work. But if you leverage something like Nextflow, you write in your scripting, basically, Nextflow, this is what I want to do. These are my job steps. I want them executed in this order. This is the inputs and outputs from each of those. And then it can go off and do those things for you and run the workflow. And it'll tell you if it stopped. If it breaks halfway, you can rerun it and it'll remember what it's done and these sorts of things. So it's really useful. Singularity is a container engine, which I don't know, too many engines. Basically, it's a way of interfacing with a container. So if you've downloaded a container from the internet and it has some software in there and you're like, I want to use the software now, please. The way that you would interface with that is Singularity. So you say Singularity run or Singularity exec this container, do this command. So like Singularity exec container name SAMtools, for example, right? So the container just by itself is not very useful. You have to use it with Singularity or Docker or some other similar bit of software. I personally prefer Singularity because the syntax is much shorter, and also you can use it on HPC because it doesn't give you admin privileges. So we don't use Docker on Cetonix, for example, because you have to have sudo to run, and we're not able to give sudo access to people. That's a terrible idea. So Singularity is the way to go.
So I guess sort of from that, is it the case? So if I had some sort of pipeline and I'd found a container to run like the first processing step, and then I had a container to maybe like, carry out some machine learning step, right? My pipeline. And it's great. I can just pull those down from even a Docker image, build them with Singularity and run them.
Yeah.
But then would I be using something like Nextflow, right, to kind of tie that process together, right?
Yeah.
So you could have these two encapsulated environments, and then I can basically say that, yeah, as you were saying, specify the inputs and outputs, and it would orchestrate that whole bigger process.
Yeah, that's exactly it. So you can tell Nextflow, I want to use XYZ containers for these various steps. Nextflow can also take Conda environments as like a software environment input. So you can specify like, all right, I want my Conda environment to have this, 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 and this. And then when you run it with Nextflow, it'll handle making the Conda environment and downloading all the packages and whatever, which is nice. I mean, typically, if you're running with bioinformatics, you typically try to avoid local installations of things where you do like sudo apt-get whatever, because there's always, oh my God, like software dependency clashes. So you try to install something and then, it disagrees with the version of R you have, but that version of R you need for this other thing that you have, and it's a hot mess. So Conda or containers help you get around that. If you're doing something that's more like just using GROMACS or one thing, then it's a lot easier because you typically only have like a few packages. You don't have like literally hundreds.
So if I'm just thinking, say, Joel, the way you're sort of describing your current situation where you have a Nimbus instance and you've kind of know the packages you're going to install, I'm guessing in whatever Python and so on. Now, just hearing your breakdown there, Sarah, I'd kind of assume that like a singularity would be kind of a good starting point there, because it's easy-ish, I imagine, to encapsulate all of that in the one container. Is that the right sort of interpretation, or is that limiting somehow?
I mean, I guess one thing to think about with containers is typically you can download pre-built ones from the internet. So there's things like key.io, which has a lot of, or bio containers has a lot of like bioinformatics containers. They don't always work off the shelf. Some of them are junk, but a lot of them are useful. So that's great. So yeah, typically we advise people don't have one container for your whole workflow, because if one part changes, then you have to change the whole container, and that can be like a big deal. It's typically preferable to have modular containers. So you have like one container for SAM tools, one container for BWA, one container for star RNA-seq aligner. Because then if you're like, oh, well, this new version of star's come out and it's way better, then you don't have to rewrite your whole container. Also, the more things you have installed in a container, the closer you are coming to dependency hell, where nothing works because all of the versions are incompatible. And that's why something like Nextflow is really good, because it handles a lot of that in the background for you. So you say, this is the container I want, and it'll go out and download them and all these things for you kind of in the background.
Yeah, I think modularization is going to be absolutely essential for us, because we did the right thing of like in the first week, wrote a list of all the tools that we suspected that we were going to be using and all their dependencies. But as we've been going, we've sort of been finding out the limitations, mainly due to like how long some of these things have been taking. And then like for some of the stuff for assembling contigs, we've like ended up changing the tool that we're using halfway through creating this pipeline, because like we've found that it's way too slow and there's another one that's quicker. So I guess having it modularized would make life a lot easier in terms of not having to go back and change everything halfway through and having to recode it all. So
yeah, totally. And like there's always new tools being released as well. Like if you're doing something like for human genome assembly, like it's so standard now that it's pretty straightforward. But if you're doing something that's a bit more like new and interesting, like plant stuff or like metagenomics or whatever, like yeah, there's new things coming out all the time. So yeah, good modularity. Single cell is like really good / bad for that. It basically sounds like there's a new tool coming out to do with the thing like every single week, and it's like, what do I pick? I don't even know.
Yeah.
Just to go off of that one, so you recommend having different environments when you're running these different sort of packages like star and SAM tools. So you could use a tool like Conda to create those different environments, and then you just switch between them as you're running each tool. Is that a suggestion?
Yeah, you could definitely do that. Yeah, so I mean, I guess it's a suggestion or like a recommendation rather than a hard rule, because there's always going to be like edge cases or whatever. But yeah, that can be quite a useful thing to do. And I've noticed that with like, for example, this ticket that I'm working on, helping them with this workflow that they have, each job step has a different Conda environment. So yeah, that can be a way to go. I mean, it depends. Like sometimes it's fine and you can package some things up together in like one Conda environment and it's okay. So I guess it depends. Like, you don't want to create for yourself unnecessary work by trying to stick to this suggested ideal, but equally, like, yeah. So I guess you have to use your judgment for what I'm trying to say.
Okay, cool. Thank you.
No worries. And I see Edric's putting in some next tutorials, which is useful. There's also on our YouTube, which you might have come across already. I think Marco did a really good. So Marco is my colleague in the supercomputing team. He's done some really good like YouTube-based trainings on Nextflow and that sort of thing. Yeah, so those things are good to check out. I do find there's a bit of a learning curve with Nextflow. It's taken me a little while to get my head around it. But also, I mean, if you've already done like, I don't know, comp sci type of thing, you're probably like, oh yeah, I get what this is about. So yeah. Cool, thanks. Yeah, another good place to find resources and things is the Australian Biocommons. So they have like just different bits and pieces of like useful documentation and like training and these sorts of things. So if you're working in that bioinformatics space, they're like useful to keep in the back of your mind that they're, you know, running things and have stuff available or whatever. Yeah. Are there more questions or comments? We're heading towards the top of the hour, so we've still got time for one or two more. Or you could get some minutes back in your day, which is always nice. I'm sure lots of us have back-to-back meetings. Yeah, all right. I'll take that as a let's have 7 minutes back or whatever. You can, I don't know, hopefully do something nice. Go stand outside. It's beautiful. I keep looking up out of my window. So yeah, okay, awesome. Good training resources there. Thanks, Edric.
No worries. And while I know I haven't had experience using Nextflow myself, thing is, you know, we'd be happy, right, to, you know, troubleshoot with you or, you know, answer any questions you have about it. Like, again, it's just I had a little more experience reading through documentation on things. Sometimes all you need is a second person to come over a set of some text, pass their eyes over it, and then point out the one thing that, you know, the one bit of connecting information that, you know, you didn't have at hand. So we're really happy to help there and assist with that kind of setup and learn alongside you when it comes to whatever workflow you're working towards. And then failing that, right, we've got the help desk and Sarah and, you know, all of that expertise there if we hit something that we can't figure out together.
Yeah, unfortunately, I'm actually going on leave from tomorrow until the end of the year. Something has come up and I can't, I've got to go. But there are other people that can help. Marco is the probably #1 Nextflow expert. He's, I don't know how he crams so much in his brain. He's just brilliant. He's a great
Clever
resource.
guy.
Yeah, it's amazing.
Awesome. Yeah, well, thanks, Sarah. It's been great. And yeah, just before we end the session, if you're able to turn on your camera, please do. And I'll just take a photo of all of us here in the session. Awesome. Thanks, guys.
Cool. Yeah, thanks everyone. Thanks for coming. Exciting to see so much bioinformatics happening at Pawsey, so it's great.
Thank you, Sarah.
Great, thank you.
Yeah, thanks for that.
Thank you.
Thank you.
Thanks. Bye.